Good morning. I'm Alexey Sivets, and I would like to welcome everyone. First of all, thanks a lot for the opportunity to participate in the conference. Let's begin. So, according to the title, our work deals with the fight against viral infections, which is of current interest extremely. We focused on preparation of modified sugars and further chemical synthesis of new modified nucleosides with potential antiviral and anti-tumor activities. As is known, structural modification of nucleosides have been successful in the search of effective antiviral agents against human immunodeficiency virus, hepatitis B and C. Modification of 2' and 3' position of ribofuranose moiety of nucleosides with different functionalities such as azido, amino group, a fluorine atom, and also nitrogen-containing moiety have been produced a variety of analogs with interesting biological properties. On this slide, is presented a number of nucleoside analogs with such structural modification. Among the presented compounds, there are compounds with both pronounced anti-tumor and antiviral activity, and in particular, antiviral activity against the new coronavirus SARS-CoV-2. Some of these compounds have been widely used in therapeutic practice for a relatively long time. In particular, AZIDO-D is the first antiretroviral drug currently actively used on as one of the key compounds of highly active antiretroviral therapy. At the same time, as a nucleoside galidesivir, which is amino-sugar derivative of adenosine, has a broad spectrum of antiviral activity against a number of RNA virus families. Is currently in clinical trials for novel coronavirus infection treatment. There were two general aims of our work. First aim was to develop methods of the synthesis of new fluoro-containing AZIDO-D-ribonucleosides of 5-alkyl 5-fluorouracils via preparation of intermediate fluorinated carbohydrate precursors from protected methyl 2,3-anhydro-D-lixofuranocide and subsequent glycosylation reaction of modified pyrimidines base with fluorinated D-ribofuranose derivatives for evaluation of their biological activities. And second aim was the synthesis of new fluorine-containing protected amino sugars as valuable precursor for preparation of 1,3-deoxy-3-fluororibitol with glycosidase inhibitor activity and novel fluorinated beta-azanucleoside with potential antiviral properties. We used for this work as a starting material protected methyl 2,3-anhydrolexofuranocide 1 obtained from readily available D-xylose by known methods, primarily by cleavage of the epoxide azido sugars 2 and 3 were obtained with high total yield in ratio of 3:1 respectively. Then we carried out the nucleophilic fluorination of the obtained azido sugars 2 and 3 using dust. An acetyl derivative of ribofuranose 6 was obtained as an universal glycosylating agent, wherein for its preparation was investigated both the acetylysis reaction and two-stage method of the synthesis. Interesting moment during the fluorination of 2-azido-D-xylofuranocide 3 with dust. We have found for the first time site reaction of migration of 5-toluene protecting group to the C3 position of uranose ring. Possible mechanism of this transformation was proposed. So the formation of byproduct 5A reduced the yield of the target fluorosugar 5 to 60%. Then protected pyrimidine 3' azido 2' fluorobeta-D-ribonucleosides 8 and 9 were prepared by the glycosylation reaction of silylated 5-fluorosyl with acetate 6 in Verbruggen conditions, followed by chromatography on silica gel. Removing protecting group of individual fluoronucleoside 8 with methanolic ammonia at the room temperature yield the protected azido-fluorobeta-ribonucleoside of 5-fluorosyl 10. Similarly, 3' azido 2' fluororibonucleosides of thymine 11 and 12 were successfully prepared by the glycosylation reaction of silylated thymine with acetate 6 in the same condition. Removing protection group of individual fluoronucleoside 11 yielded to 3' azido 2' fluorobeta-D-ribonucleoside of thymine 13. Synthesis of similar 2' fluoro 3' azido-D-ribonucleosides of 5-propyluracil under the same condition using acetonitrile as the solvent failed. The reaction did not lead to the desired nucleosides 14 and 15. Then replacing the solvent with dichloroethane allowed nucleosides 14 and 15 to be obtained in similarly low yields after chromatography on silica gel. At the same time, the use of 1-brom sugar 16 in the condensation reaction using dichloroethane as a solvent in the absence of Lewis acid as a catalyst resulted in an exclusively alpha isomer of the protected nucleoside. Removing protection group of individual nucleosides of 5-propyluracil 14 and 15 yielded corresponding the protected ribonucleosides 17 and 18. Protected 2 azido 3' fluorobeta-D-ribonucleosides of 5-fluorosyl 19 and 20 was prepared by the glycosylation reaction of silylated 5-fluorosyl with methyl ribofuranosides 5 in acetonitrile, removing of protecting groups of individual beta nucleoside of 5-fluorosyl 19 yield corresponding ribonucleoside of 5-fluorosyl 21. Second aim of our work was the synthesis of target 3-fluorosubstituted 1,4-dideoxy 1,4-IMINO-D-RIBITOL derivative that was accomplished in 7 steps from methyl 5-benzyl to 3-anhydrodeoxyfuranozide 22. Tazolate 23 was prepared using nucleophilic cleavage of epoxy 22, followed by treatment of fluorosugar with paratoluene sulfonyl chloride. The ribitol derivative 25 was synthesized from tazolate 23 by acidic hydrolysis of methyl arabinoside, followed by reduction of intermediate 5-benzylated aldofuranose 24 with sodium borohydride. Key intermediate methylated sugar 28 was synthesized following by the forming of the 1,2 epoxy cycle, obtaining epoxide sugar 27 followed by its methylation. Protected 3 fluorinated pyrrolidine derivative 30 was prepared via encyclization of intermediate methylated sugar using benzylamine and can be used as a valuable precursor for preparation of novel fluorinated beta-nucleosides 33 with potential antiviral properties. Also, new IMINO sugar 32 with potential antiglycoside and antiviral activities was obtained by the protection of debenzylated 1,4-IMINO-D-RIBITOL via catalytic hydrogenation. So our summary will be next. synthetic multi-step approach to protected fluoro-containing AZIDO-D-RIBOFURANOSES were developed from D-cellulose using nucleophilic fluorination of 5-phosphorylated AZIDO-D-RIBONOSIDE and xylofuranoside with dust. We have found for the first time that migration of 5-toluene protecting group proceed during fluorination reaction of 5-phosphorylated to AZIDO-D-xyloside with formation of 3-phosphorylated D-xyloside. Possible mechanism of this transformation was proposed. A series of novel azido and fluorobeta and alpha D-ribonucleosides of 5 substituted pyrimidines were synthesized from fluorosugars for evaluation of their antiviral and anti-cancer activities. Nerve peculiarities of glycosylation reaction of accelerated 5 substituted uracils with fluoro-containing AZIDO-D-RIBOFURANOSE derivatives have been established. Synthesis of protected 3-fluorosubstituted 1,4-IMINO-D-RIBITOL derivative was developed in 7 steps from methyl anhydro-D-lixofuranoside using encyclization of intermediate syntone containing the terminal epoxy cycle. Novel 3-fluorinated dipyrrolidine derivative with potential antiglycosidase and antiviral activities was synthesized from debenzylated D-ribitol precursor. The protected 3-fluorosubstituted 1,4-IMINO-D-RIBITOL was synthesized as valuable intermediate for further preparation of 3-fluorinated purine carboxynucleus nucleosides with potential anti-SARS-CoV-2 activity. Synthetic methods developed for fluorinated sugars and nucleosides are of practical interest for designing novel pentafuran and other nucleosides, and there are also phosphoimidate derivatives with potential biological activities. That's all. Thank you for attention.
Thank you for the speaker. Anybody wants to make questions?
Can I again?
Yeah.
Thank you. Thank you, Alexey. This was a quite big job. I would say a huge job. Organic synthesis is not easy, right?
Yes.
Please tell me, did you test these compounds? You planned them to have antiviral or some kind of antimicrobial activity?
It's now in our plans. But we have not tested these compounds now.
Not yet, right?
Not yet, right.
Okay, okay. I think there are plenty of interesting compounds for these activities. Thank you.
Алексей, скажите, пожалуйста, покажите слайд 4, пожалуйста, или 5 там, ну, они... Тогда следующий, пожалуйста. Вот там, где у вас, ну вот этот можно там. Вот у вас соединение 2 и 3. Во-первых, у меня первый вопрос. Удалось ли вам их разделить? Ну, судя по реакциям, удалось, да?
Yes, yes. This compound,
и второй, тогда второй вопрос, чисто такой по установлению структуры. Как вы отличаете структуры вот соединение 2 от соединения 3? Какими методами вы доказывали и что вы видели?
Using nuclear magnetic spectroscopy, first of all.
Пожалуйста, конкретно скажите, чем отличаются, вот, ну, если вы говорите ЕМР, значит, чем вы, как вы видите отличие этих соединений?
Chemical shifts of signals of protons of these compounds and constants, coupling constants.
Не помните, да?
Okay. Any other questions? I have one question. You told that these compounds are potential anti-SARS-CoV-2 drugs. Say,
No,
So
maybe as analogs of presented here compounds.
mhm. And will you test them on anti-SARS-CoV-2 activity or not?
It's in our plans, but we have now not tested these compounds.
Okay, thank you.